The next presentation is Remote Ischemic Conditioning Stimulates Proangiogenic Mechanisms in the Intestine During Experimental Necrotizing Enterocolitis, and it is being presented by Niloofar Ganji from the Hospital for Sick Children at the University of Toronto.
Good morning, everyone. Thank you very much for the opportunity to present our work. I'm very grateful for being here. Remote ischemic conditioning is a treatment strategy that involves applying brief cycles of non-lethal ischemia and reperfusion to an extremity that then leads to the protection of distant organs, such as the intestine, against ischemic damage. The Pura Lab has recently shown in our preclinical studies that remote ischemic conditioning, when applied in early stages of the disease, reduces the intestinal damage to NEC and prolongs survival by enhancing intestinal microcirculation. However, the precise molecular mechanisms underlying the beneficial effects of RIC are not clearly understood. In this study, we investigated the hypothesis that the mechanism of action of RIC during experimental NEC involves the modulation of angiogenesis in the intestine. As a brief background, angiogenesis is the formation of new blood vessels from pre-existing vasculature, a complex process that is mediated by vascular endothelial growth factor, or VEGF, along with its receptor, VEGF receptor 2. There are several models of vessel formation. These include the sprouting of endothelial cells, or sprouting angiogenesis, as well as the recruitment of endothelial progenitor cells, or EPC, that then differentiate into endothelial cells. In our experimental model, NEC was induced from postnatal day 5, or P5 through P9, by hypoxia, oral lipopolysaccharide, and gavage feeding of hyperosmolar formula. Breastfed littermates served as controls. RIC was administered on postnatal days 6 and 8 by intermittent occlusion of hind limb blood flow with a tourniquet. And to investigate the role of angiogenesis in the mechanism of action of RIC, NEC pups receiving RIC were given a VEGF receptor 2 blocker. Starting with our first results, we saw, consistent with the literature, a downregulation of VEGF in our NEC model, which is shown in red compared to the breastfed controls. However, following remote ischemic conditioning, both the gene and protein expression of VEGF were upregulated. So you can see the qPCR expression of VEGF on the left-hand side and immunofluorescence staining on the right-hand side. In the immunofluorescence staining, we looked at microvessels within the villi using antibodies against VEGF, that is shown in red, and antibodies against CD34, that is shown in green. And CD34 is a cell surface marker that marks endothelial progenitor cells, as well as endothelial cells that are resident within vascular walls. On this slide, you can see the quantification of the immunofluorescence. So we took each villus of interest, we divided it into 4 equal sections from 0 to 25, 25 to 50, 50 to 75, and the top 25% of the villi. And we saw that, surprisingly, RIC promoted the expression of VEGF by CD34 positive endothelial cells, particularly at the top of the villi, but not at the crypt base, not at the bottom 25%. And when we administered the VEGF receptor 2 blocker to NEC pups receiving the RIC stimulus, we saw that the beneficial effects of RIC in reducing intestinal damage and inflammation were abolished. So you can see the histological staining images, as well as the scoring with a grade 2 or above indicative of NEC, and you can see the qPCR expression of pro-inflammatory cytokine interleukin 6 on the right side. After showing that RIC does modulate angiogenic mechanisms in the intestine, our next question was, how is this proangiogenic signal of RIC conveyed from the hind limb to the intestine? And to answer this question, we hypothesized a role for stromal cell-derived factor 1 alpha, or SDF1 alpha. This is a humoral chemokine with proangiogenic activities that's particularly involved in homing, retention, and proliferation of endothelial progenitor cells. The proangiogenic mechanisms of SDF1 alpha are stimulated through the activation of VEGF, as well as the activation of CXCR4, which is the SDF1 alpha receptor, as well as the recruitment of endothelial progenitor cells to ischemic sites such as the intestine during NEC. To investigate the role for SDF1 alpha, we looked at 2 tissues, the ileum as well as the hind limb that was receiving the RIC stimulus. And we saw that while in the intestine, the SDF1 alpha protein expression was elevated with no change in gene expression. In the hind limb receiving the RIC stimulus, both the protein and gene expression of SDF1 alpha were upregulated. And this suggests that following RIC, SDF1 alpha may be mobilized from the hind limb to the intestine. And next, we wanted to see whether with SDF1 alpha treatment, we're able to mimic the protective effects of RIC in the intestine during NEC. So we looked at intestinal histological injury with the same scoring system, grade 2 or above defining NEC, as well as the qPCR gene expression of pro-inflammatory markers, interleukin-6 and tumor necrosis factor 1 alpha. And in fact, we saw that both the effect of histological injury and inflammation were reduced with SDF1 alpha treatment. And finally, to look at the downstream mechanisms activated by SDF1 alpha, we looked at the gene expression of its receptor, CXCR4, as well as the gene expression of VEGF, and saw that, in fact, both were upregulated. And we also saw a very similar pattern of upregulation of VEGF by CD34 positive endothelial cells within the villi microvasculature, particularly at the top of the villi, not at the crypt base. And this is important because we've shown previously that in NEC, it's the enterocytes, particularly at the top of the villi, that suffer the greatest ischemic damage. So in summary, following a remote ischemic conditioning, SDF1 alpha may be mobilized from the hind limb to the intestine, where it then activates VEGF as well as other angiogenic growth factors and mobilizes endothelial progenitor cells, leading to endothelial differentiation. There's a large team behind me. I would like to thank each and every one of them, as well as you, for your attention. Thank you again for the opportunity to be here, and I'm happy to take questions.
This paper is now open for questions.
It's a very elegantly designed study, and I congratulate you on such interesting results. What's the next step forward for your lab using this data?
Thank you. I was hoping someone will ask that. So
I wasn't paid.
Yes, thank you. So the next step is big and exciting. In fact, we are currently, just before I was leaving for San Diego, we got the final RB approval for an international multi-center study, a phase 2 feasibility study looking at RIC in human babies with NEC. So this involves sick kids in Toronto. It involves a few other hospitals in Ontario in Canada, as well as a few centers in the US and many centers in Europe. So we're very excited for this upcoming study, and this is our first, we've completed a safety study looking at the safety of RIC in human babies with NEC in sick kids. So this is our next step before we proceed with the efficacy trial, just because we need to answer many questions related to feasibility of RIC before we move on. But as we are proceeding with the translational aspect, we continue our science studies, of course, to understand better the mechanism of action of RIC, because we really believe that it works by targeting the microcirculation, and if we're able to catch these babies early on when the defects in microcirculation start to happen before they proceed to a more advanced stage of NEC. And if we can do that with RIC, then that may be a novel treatment for NEC that may change the management of this disease worldwide. So that's what we're hoping for next.
Congratulations, that's wonderful, truly bringing the bench to the bedside. Thank you.
Thank you very much.